So, hello doctors. Topic of discussion is acquired immunity. I am Dr. Amin Bharti Porwal. So, types of immunity. Number one is innate immunity, or called as non-adaptive immunity. It is non-specific defense, and relatively sometimes they are specific by NK cells. So, NK cells are involved in innate immunity. And second variety is acquired immunity or adaptive immunity, in which there can be naturally acquired, like active immunity. They are usually infection through infection. After getting infection, we can have cellular immunity or humoral immunity, or in the passive immunity, transfer of antibody from mother happen. IgG via placenta and IgA via breastfeeding. Artificially acquired immunity, it can be active immunity like vaccination, or it can be passive immunity, again by vaccination. So if you are giving attenuated antigen, it is active immunity, and if you are giving antibodies, it is passive immunity. Now, objective of today's lecture is cellular or humoral immunity. Step 1. Step 1 is antigen recognition, processing, and presentation. Step 2 is activation and proliferation of the T cell. Step 3 is elimination of the invader. So, number 1 is antigen recognition, processing, and presentation. Antigen presenting cells are macrophages, they are dendritic cells and B cells. So, what are the five major steps during antigen presentation? Number 1 is ingestion of the antigen. Number 2 is digestion of the antigen. Number 3 is fusion of the vesicle. Number 4 is binding of the peptide fragment to MSC molecule. And number 5 is incorporation of antigen MSC complex into the cell membrane of antigen presenting cell. Now, class 1 MSC molecule bind to antigen fragment. For that, you need to remember the diagram first. So there is alpha 1, alpha 2, alpha 3, and beta 2M. This is the plasma membrane. So where is the antigen binding groove? Where is? Answer is between alpha 1 and alpha 2. So antigen bind with which space in the major histocompatibility complex class 1? Answer is between alpha 1 and alpha 2. Now step 2 is activation and proliferation of the T cell. So interaction between antigen presenting cell and T lymphocyte is called as immune synapse. Between antigen presenting cells, you can see here that between alpha 1 and alpha 2 there is antigen fragment, and this TCR is T cell receptor heterodimer. Why heterodimer? Because you can see in this diagram there is alpha beta chain. It is called as variable chain, and in the constant region there is a bond called as disulfide bond. Now, class 1 antigen and class 2 antigen. Class 2 antigens are present in professional antigen presenting cells. Professional, including B cell and dendritic cells. And class 1 antigens are found on all nucleated cells. So, class 2 are found in all nucleated cell. Class 1 are found in all nucleated cells. Class 2 antigens are present only on professional antigen presenting cell. They need to present antigen from your body to the T cell, from T cell receptor. So you can see here that class 2 MSC bind with TCR with the help of CD4, and class 1 MSC bind to TCR with CD8. Now MSC1 are coupled to peptide fragments from protein which are synthesized within the cells, which are not tolerant and recognized by the T cell. So like mutant protein, like viral proteins, which are not tolerant. So they are present on, class 1 is present in all nucleated cells. But some cells are not tolerant. They are not good for the body. Like viral proteins, like mutant proteins, they are recognized by class 1 MSC, and they are recognized by the T cell. So digestion of these proteins occur in the complex of proteolytic enzymes called as proteosomes, and the peptide fragment bind to MSC proteins in the endoplasmic reticulum. This was about class 1 MSC. Now MSC2 interact with the peptide product of the extracellular antigen such as bacteria. So class 1 is the own, but they are mutant. Class 2 are for the bacteria because they are antigen to the body. So for that we need class 2. So you must be very much clear about class 1 and class 2. Now the main scene start coming. So what happens when antigen comes in your body? They are taken by the macrophage, which are antigen presenting cells. They have MSC2 and they need to present to the T lymphocyte. So this is T lymphocyte having T cell receptor and CD4. So MSC class 2 needs CD4, MSC class 1 needs CD8, and macrophages will process the antigen, will present the antigen to the T lymphocyte, so they need to talk. So normally when we talk, it is via chemical, but when macrophage talk, they talk via some chemical that is called as interleukin 1. So interleukin 1 is secreted by macrophage and going to stimulate T cell, T lymphocyte. So this type of stimulation is called as what? Autocrine or paracrine? This is called as, you can see here, it is called as paracrine stimulation. So interleukin 1 is released by the macrophage and going to stimulate T lymphocyte on TCR with CD4, and this stimulation, because one cell is stimulating other cell. So what is this called as? It is called as paracrine stimulation. You can see here that this is called as paracrine stimulation. Now after paracrine stimulation, T lymphocytes will be stimulated. And when they are stimulated, they will release interleukin 2. And this interleukin 2 is going to stimulate whom? To whom they are going to stimulate? Nobody but themselves. So interleukin 2 are released by T lymphocytes and are going to stimulate themselves, and this is called as autocrine stimulation. So that was called as paracrine stimulation. This is called as autocrine stimulation. So now this is stimulated T lymphocyte. Now it can cause clonal formation, it can cause inflammation and delayed hypersensitivity, and it is activated T lymphocyte. Now, this activated T lymphocytes, they can cause cytokine-induced activation of the plasma cell into B lymphocyte. But through interleukin 4, interleukin 5, so now activated T lymphocytes are going to release interleukin 4, interleukin 5, and they will activate whom? They are going to activate the B cell, and now they become antibody-producing B cell. So these plasma cells can produce antibodies. This is called as humoral immunity. Till antibodies are not formed, it was cellular. But when antibodies are formed, it is called as humoral immunity. So who is activating that? Interleukin 4, interleukin 5. These activated B lymphocytes will become antibody producing cells. And also, not only this, interleukin 2 are also going to form cytotoxic T lymphocyte. And now they bind with MSC class 1 to cause cell death. This is cellular immunity. So that was humoral immunity. This is cellular immunity. And this is called as elimination of the invader. Invader is annihilated. Now, after that, antibodies are formed. We must know the basic structure of antibody. We can see here, this is the heavy chain. Now we can see here, this is the heavy chain CH1, CH2, CH3. And between them, we have disulfide bond. And that chain is called as light chain, VL and CL. L means light, H means heavy. The upper portion is called as FAB, antigen binding. FAB is concerned with the antigen binding, and the lower is called as effector portion of molecule, and they also have a hinge on which we have disulfide bond. Now let me summarize everything. So whenever bacteria, virus or tumor enter in your body, neutrophils are activated. They will cause respiratory burst, and they will cause all this superoxide dismutase and all, and they will try to engulf the antigen. And after that come the main hero, the macrophage, and they can act like an antigen presenting cell. And antigen presenting cells can go in two different directions. They will first stimulate T lymphocyte, and if they form Th2, if they stimulate Th2, they will stimulate B cell to form plasma cell, and plasma cell will form antibody. On the other hand, if they activate Th1, they form cytotoxic lymphocyte, and they are going to engulf the antigen. Now, let us discuss some MCQ. T cell receptor engagement with antigen MSC complex is a crucial step that triggers T cell differentiation, T cell death, T cell activation and proliferation, or T cell migration. Now we know answer is T cell activation and proliferation. Step 2. Which signaling molecule is the central to T cell proliferation and often used in the therapy to promote T cell expansion? We know the autocrine stimulation by T lymphocyte, and that is interleukin 2. Third, what is the primary function of helper T cell? During T cell proliferation, we know to activate other immune cell, either they can include Th1 or Th2. If Th2, then B cell antibody. If Th1, they form cytotoxic T cell. The last and the most important. The expansion of T cell population is tightly regulated to avoid excessive immune response. Which type of T cell play a role in controlling this regulation? The negative feedback, regulatory T cells. Now let me summarize. There are 4 subtypes of helper T cell. T helper 1 secrete interleukin 2, gamma interferon, and concerned with the cellular immunity. T helper 2 secrete interleukin 4, interleukin 5, and interact with the B cell in relation to the humoral immunity. T helper 17 are produced in response to the bacterial infection, produces interleukin 6, interleukin 17, and help in recruiting neutrophils. T regulatory cells produce interleukin 10 to dampen T cell driven response. Thank you.